we're getting into some very specifics. We're going to go a little bit through mutation, and then we're going to get into some specifics of why microbes are particularly variable, and why they evolve so quickly. So we'll get into that in a little while. Next week, then, we'll have a pretty busy week next week. We'll go just briefly, I'm going to touch on DNA technology. I usually go through that a little bit in this class, but I don't need to go through that too much since you all have cell and molet, and many of you are more about. I'm just going to go through that very lightly. But it's important to understand some of those methods before we get into things like microbial diversity. We'll go through diversity for a little while, and then we'll have a review session again following Tuesday, and then another exam. So this is happening really fast. Our next exam, the semester's almost over, it's crazy. Our next exam is the day after Halloween. So sorry about that. Don't bring guests in. Okay, so today, this is genetic variation, and you guys have seen this before. I've shown this to you a couple of times. This is the tree of life. And I've told you to take home a few things from this image. One, almost everything on the tree of life is microbial. Almost every living thing, in terms of absolute numbers on this planet, is a microscopic organism. But also, they're just amazingly diverse. And so what we're going to go through today is the mechanisms of that diversity. Microbial organisms, particularly bacteria and archaea, don't follow the same rules of natural selection and evolution that macroorganisms do. They share DNA freely amongst each other, and I'm going to explain that to you. So making the species concept with respect to microbes pretty tricky. And I've touched on that before in this class, but by the time you're done with this lecture, I hope you understand what I mean by why we have a hard time with this species concept with respect to microbes. So when we think of a species with macroorganisms, what do you guys think? How do you define them? How would you define why are you a species and a different species than your dog and then an oak tree? What's the biological species definition? Mike, you ready?
Reluctantly able to mate with one another and
Sure, that's the simplest biological definition, is that they're able to mate with each other and produce viable offspring. Viable offspring that can then go on and mate again and produce more viable offspring. And so with bacteria, though, and archaea as well, that gets very tricky. They share DNA freely across many different species lines. They'll do things that's comparable to us sharing our DNA with, say, a dog. It's that phylogenetically different, or even a tree or a plant. It's that different. And we're going to go through those mechanisms today. So this is just a reminder of putting it in the big context of just the vast diversity of microorganisms and how they don't play by the same rules we do with respect to their genetics and their reproduction. Okay, so let's think about the sources of variation. What I'm implicitly saying here is that sources of genetic variation lead to this diversity, lead to natural selection and evolution. We're going to go for a little bit in the beginning through mutation. A lot of that will be a review for you, but just mutation in the simplest sense. And then we're going to get into the second half of the lecture is going to be on these three methods of what I call lateral gene transfer. People call it lateral gene transfer, people call it horizontal gene transfer. And forgive me, when I'm talking fast, I use those terms interchangeably and I don't want to confuse you. They're the same thing. Horizontal, lateral, all the same thing. But those three methods are conjugation, transformation, and transduction. And we'll go through those in a fair amount of detail. Okay, so mutations. Mutation is just a stable heritable change in a piece of DNA. It can be spontaneous or induced. And we so often think of mutations as a deleterious thing. Even when I'm kind of running through my notes, getting ready to come lecture to you guys on this topic, I'm thinking to myself, oh, well, if this mutation happens, then that's a problem. But obviously we know that's not really the case. None of us would be sitting here without a whole bunch of beneficial mutations over the last billion years. So mutations, in many cases, we think of them as deleterious and causing a problem. Not always. Okay. In the grand scheme of populations through time, they're obviously pretty beneficial. Okay, so let's spend a little bit of time just talking about spontaneous mutations, and then I'm going to kind of define for you what an induced mutation is. So a spontaneous mutation is going to arise without exposure to some other agent, without exposure to UV, some sort of intercalating agent, or something that binds and messes up your DNA. All right, it's going to result in an error. Sometimes those errors are very problematic, result in a big change. Sometimes there's no real change at all. Okay, and so one example of that is a point mutation called a tautomerization. And this is an example of a point mutation. Anyone know what I mean by a point mutation? What do you, just intuitively, what do you think I mean by a point mutation?
It's literally just one base pair switching.
Beautiful, just one base pair. You should do this, just one base pair, right. All right, so in tautomerization, what's happening is you just have rare forms of your nucleotides that bind incorrectly. So you remember that A's bind to T's, A's bind to T's, G's bind to C's, etc. But when you have these rare forms that are just slight shifts in the molecule, slight shifts in the nucleotides, you'll end up with a T bound to a G, or an A bound to a C, etc. And so what ends up happening is here you have an example of a parental strand of DNA, and in this case you have G and C bound to each other, just like we would expect. And then as that DNA replicates, you have this basically strange form, or tautomeric form of guanine, such that it binds to T instead of to C. All right? So here, and then on the other strand, the C and G bind together just like they normally would. But this strange form of guanine, instead of binding, cytosine binds to thymidine. All right? And so in that one spot, you just have one nucleotide off, and it doesn't seem like a terribly big crisis. All right, for this little strand. But when you think about it through generations of replication, you're going to end up transferring that down and create a permanent mutant, such that here, when the normal strand that didn't have a mutation, you have its first progeny, and then it replicates, and it continues to be a wild type on down the line. But on the strand that had tautomerization, you end up with G and T there instead of G and C. This strand is going to divide and replicate itself, and then when it replicates itself up here, it goes back to binding to the C in this case. So this was just one rare incidental event. It goes back to binding to the C, you end up with the wild type. But on this other strand, the pink strand, now you have a T down there. So you're not going to have a G up above, you're going to end up with an A up above. And so now you have a mutant at that exact base pair spot, that point. And that can get carried on. And in a couple of slides, we're going to talk about point mutations don't seem like such a big deal, like such a big mutation event. And it depends where they are. And we'll talk about that in just a couple of slides. Okay. You can also have insertions and deletions. So as DNA is being replicated, you can have something called slippage. And slippage is when either the parental strand or the replicating strand just makes a little bubble and slips away from the replicating two strands together. And what it does is it ends up inserting or deleting a couple of nucleotides. So in the case here, a slippage leading to an insertion, where you have slippage in the new strand. So you have a new strand being laid down, and in this case it forms a little bubble, and it slips off to the side, and you end up inserting one nucleotide because the G and the T didn't get replicated properly. And you end up inserting one more nucleotide. So ideally, these are perfect opposites of each other, but instead you end up with 1, 2, 3, 4, 5, 6 T's in a row. And down here on your parental strand, you have 1, 2, 3, 4, 5 A's. So basically what we've done is we've inserted an additional thymidine where it didn't otherwise exist. All right. And if you have slippage in the parental strand, you're going to end up with a deletion. So in this case, you have the parental strand forms a bubble and falls down. So then these nucleotides don't get read. And you end up basically omitting them as you replicate the parental strand into your new strand that will get replicated, and that's a deletion. spontaneous mutations, obviously there's a lot of other ways that happen too. You can also induce mutations. There's a lot of different ways to induce mutations. I don't expect you guys to memorize this table. Anything that binds DNA could potentially create a mutation. And then also other physical factors like UV light and X-rays. UV light creates pyridine dimers. I'll show you that in just a second. Things like acridine orange is an intercalating agent. A lot of microbiology labs use acridine orange as a stain. I do in my lab. And the reason that stains the cells is it binds up to the DNA and then it fluoresces. You can look at it under the microscope. It's actually very pretty. It glows bright orange. It's very pretty. Okay. UV causes thymidine dimers. And in that case, what you have is if you have T's in a row of your strand, of your nucleotide strand, and you end up with binding across between the T's that didn't otherwise exist, causing TT dimers. And it makes your strand very rigid. It's not going to function the same way. It's not going to replicate itself the same way. Messages aren't going to be transcribed the same way. All right. Let's step through this table. So let's think of the effects of some of these mutations. So a minute ago or so, I talked about point mutations. I said, well, sometimes they matter, sometimes they don't matter so much. All right? But let's step through it. And when it matters, it depends on the placement in that 3 codon position. So remember, each 3 codons codes for a particular amino acid. All right? And so it depends. And if it's at the beginning of that 3-place codon, then it might cause quite a problematic mutation and lead to a different amino acid. If it's at the very end of that chain, it may not cause such a problem. And who remembers the concept that I taught you guys that you learned that is the framework for why that's the case? A couple of lectures. There's a word I taught you guys, a concept I taught you that in the 3rd position, wobble, good. In the 3rd position, it doesn't matter so much. You could get a bunch of different, you could have a bunch of different nucleotides there, and you're still going to get the same amino acid because the genetic code is very redundant. All right, so let's step through this table. All right, so if you have a base substitution, which is just basically a point mutation, here at the top line, and this is obviously from the textbook too. Here on the top line, you see a nucleotide sequence. Every 3 nucleotides codes for an amino acid. All right, the first one, ATP, goes to the thymine. This one codes for an amino acid. And if we just do a base substitution, at the 3rd position of this 2nd amino acid, it goes from ACC to ACA, you still get threonine. So you're not going to end up with a different protein. It's not going to alter the protein morphology and protein structure. All right, we call that a silent mutation. Nice and intuitive, silent. It doesn't really change your protein. All right, but if we get that base substitution at a different location, you can end up with a missense mutation. And a missense mutation actually changes the amino acid because in this case, 1,2,3 methionine, 1,2,3 threonine, 1,2. That base substitution happened at the 2nd position in that 3 codon set. And then you ended up with cysteine instead of serine. All right, and we call that a missense mutation. So now we have an amino acid that's been shifted, and it may affect the folding of that protein, it may affect the activity of that protein. A nonsense mutation is when you end up with a nucleotide swap and it changes it to something like a stop codon right in the middle. Stopping transcription right there, right in the middle. All right. And one of the most changing of mutations is a frame shift mutation. So there, I want to use the word deleterious, but the biggest change can come from a frame shift mutation. And that's when you have either an insertion or a deletion. And I'll just show you kind of how insertion and deletion can occur. If you have an insertion or a deletion, what it's going to do is, remember, we're reading this in sets of 3. Okay? ATG gives you methionine, ACC gives you threonine, ACC gives you serine. If we insert a nucleotide there, then every downstream from that is gonna get moved over by one place. Every one of them after that is gonna be different, okay? You just shifted the whole thing down, even just by one nucleotide, and it completely alters the amino acid sequence of that protein. All right, hence calling it frame shift. We've moved it down, and now you'll see they've highlighted nicely for us in red. You have a completely different sequence of amino acids following that mutation event, either that insertion or that deletion. Shifted everything down. All right, so that's going to be a pretty profound mutation in the sense that now the protein's not going to function. It has a completely different amino acid sequence. No telling what it's going to look like. All right, we can reverse mutations. They have the ability to heal themselves. You guys are going to do this in lab in a week or two. Where you're going to put your plates, put your cultures under UV light, expose them to UV light, and then you're going to have a set of those plates that gets left in the dark, and a set of them that gets left in ambient light, or I think we have a lamp for you. We'll talk about how we can reverse mutations too. All right, so just simple reversing of mutations. Here you had a parental strange, you had a forward mutation that switched threonine to proline, and basically what happened is it was, let me see, ATG, ACC, UTG, and then C. So in the first put on position, it switched from threonine to proline, and then it just switched back. And that can happen. It can totally happen. It just switched back and it basically fixed itself. All right, and again here you switch serine to cysteine. In this case, you can have basically a reversion that doesn't necessarily perfectly heal the mutation, but it lessens the impact of it, such that in this case, we switch from threonine to proline by switching this nucleotide. Another nucleotide comes in and changes it again, but instead of switching it back to threonine, it switches to leucine. Now, I didn't make you guys remember all the structures of amino acids, and I haven't had to memorize those since I was about your age, but you know that you have polar and non-polar amino acids, and some of them look very much alike, and they're going to behave biochemically very much alike. So in this case, they're saying, you started with a polar amino acid, went to non-polar, and then the reversion took it back to a polar amino acid. So the morphology of the protein may have remained somewhat intact. They called it a pseudo-wild type. Alrighty, so let's talk quickly about suppressor mutations. So in this case, we had a forward mutation that was a frame shift. We removed a G there. And so you ended up with a completely new amino acid sequence following that frame shift. But what if something comes along later in the next generation and does an insertion or deletion, etc., such that it returns back and it reinserts something such that we can reset the frame shift. It either removes or inserts such that you can basically reset it. Think of yourself as moving along a tape and reading a tape, and you get lost in your spot, but then something else comes in and puts you right back where you were. And so that's going to be a suppressor. Okay, questions on any of that? Alrighty. So let's think about this genetic variability. Okay, obviously, I'm assuming everyone is comfortable with the fact that mutations generate genetic variability. And this absolutely happens in microbial cells all the time. It's happening in our bodies as we speak. But another way to think about genetic variability is recombination. Creating recombinants. For those of you that have had genetics, you're like, yeah, right, whatever. Those of you that maybe don't remember your genetics or that fundamental biology, a recombinant is just taking an individual organism and recombining its own genetic material. Macroorganisms, eukaryotes, don't do that very often relative to bacteria and archaea. Bacteria and archaea do it constantly. They recombine their DNA with that of other cells or little chunks of nucleic acid that are just floating in the environment all the time. And they take it up through a couple of methods we're going to go through in just a minute, and they incorporate it into their own chromosome, thus recombining their chromosome, creating a new sequence that they did not previously have, new genetic variation that they did not have. So we're going to talk about the methods for that now. There's something else I wanted to say that come in. Okay, so what we're talking about is horizontal gene transfer. define these terms for you. Conjugation, transformation, and transduction. These are three hugely important words. This principle is hugely important to microorganisms. It's the fundamental reason that microbes evolve so differently in the way they do, so differently from eukaryotes and macroorganisms and the way they do. It's also the principle that's behind things that we take for granted, like antibiotic resistance. Okay? Happens this way. So when we go through this, it'll help you understand that. It's also why we're able to take a bunch of microbes and put them in a toxic oil spill down in the Gulf, and they will adapt and change rapidly, decaying and decomposing some food and organic matter, etc. Okay, because they can recombine so easily through these methods. So just in the grand sense of how this can play out is you have a recipient cell and some kind of donor DNA. The origin of that donor DNA we're going to go through in the next few slides. Okay? We go through lateral gene transfer, horizontal gene transfer, and somehow this cell takes up that recipient DNA. All right? That recipient DNA can go through a couple of different pathways once it gets on the inside. All right? It can go through integration into the host chromosome. It's perfectly recombining with the host chromosome. And you end up with a completely, and then that cell reproduces, and you end up with a perfectly healthy, viable, stable recombinant. All right? It can also, in the case of a plasmid, it can get incorporated in as a plasmid. Yes, cells can take up little plasmids. I remember the point I was going to get to. I'll get to it in just a second. They can take up little plasmids. That cell can reproduce itself, and that plasmid can reproduce itself autonomously in many, many cases. And then you end again with a stable recombinant. All right? In some cases, the donor DNA doesn't self-replicate. Okay? And you can even have cases where you have host restriction. Anyone know what I mean by host restriction? Anyone know what I mean by host restriction? Who here knows what a restriction enzyme is? That's with the DNA, absolutely. So we use restriction enzymes in the lab all the time. You guys are a little snoozy on me this morning. Who here has used a restriction enzyme in the lab? Wow, really? Okay, well, we'll talk about that. So restriction enzyme is an enzyme that naturally occurs in a lot of different cells, and the point of a restriction enzyme is to cut up pieces of DNA. And in a completely natural environment, this is going to be understandable as a cell takes up DNA and then wants to recycle it and use it. Okay? So it could conceivably take up a piece of DNA, cut it up into a million little pieces, either use it energetically, DNA has a lot of energetic value, nutritional value, or cut it up and use it for its own DNA synthesis. Anyway, point being, it's no longer a stable piece, and you don't end up with stable recombinants. So the point I was getting to earlier, and that some of you I know have done, but maybe not as many as I thought, is these are the underpinnings of a lot of molecular biological techniques. So, for instance, we exploit these methods, like we exploit transformation, that I'm going to describe to you in just a second, so that we can create recombinants in the lab. A lot of the genetic experiments, microbiological experiments, etc., are done using recombinants and using these principles. I do principles in the lab all the time where I'm just transforming healthy cells and getting them to take up a plasmid so that I can manipulate that cell into doing something I want it to do. So this is heavily, heavily practiced in molecular biology, and certainly in any of the pharmaceutical industries around here that you guys might end up looking at. All right. Okay, so let me remind you what a plasmid is. Plasmid is a separate, autonomous, little bitty ring of DNA. Okay? A little double-stranded ring of DNA. Autonomous in the sense that it is not normally, in some cases it is. In the case of an episome, in the case of an episome, it is integrated into the host chromosome, but usually not. It's usually a separate little piece of DNA. Bacteria and archaea tend to have tons of these things. And what we're going to go through in a lot of detail today is conjugative plasmid, the F plasmid. And we'll go through that when we go through conjugative. what that little plasmid does. But plasmids tend to carry non-essential genes. All right, you guys learned about this for the first test. Antibiotic resistance is frequently carried on a plasmid. Okay, and cells can transfer plasmids freely amongst each other. All right. And the F plasmid is a special case, and we'll talk about that. So let's start with conjugation. Of those three methods, let's start with conjugation. So conjugation is a case of cell-to-cell contact directly for the purpose of creating a recombinant. Okay? Cell-to-cell contact where one cell will contain an F plasmid. It will literally build a big long tube and reach out and touch a recipient cell. And in the process, replicate its F plasmid, send it through the tube, and give it to the recipient cell. The tube is cleverly named a sex pilus. So what you have here is two cells touching each other through a tube and sharing genetic information. Shocking thing is, this is not sexual reproduction. Okay? Bacterial cells do not sexually reproduce, but they do share DNA. And this is lateral gene transfer. Sexual reproduction is vertical gene transfer. This is lateral, horizontal gene transfer. So, the donor cell will have one of those F factors, one of those F plasmids, and then we'll call that cell F plus. Okay. That F plasmid will code for all sorts of genes, like replicating itself, like creating the sex pilus, like excreting a bunch of enzymes so that the sex pilus can contact his buddy there and actually insert that the replicated F factor into the new cell. All right. So the F factor is the plasmid. And it contains the information for the sex pilus, but also it contains genes for secretion systems. So it can secrete that newly replicated F factor. Okay. And relaxases, enzymes that will actually relax that strand of DNA so that it can slip through that tube. Okay. Things like coupling proteins. Yeah.
What triggers that tube to be made?
I'm sorry?
What triggers that tube to be made between two bacteria?
Would you say how thick is it?
No, what triggers
What
that to be
triggers?
made?
Cool question. I don't know. I bet that's a really great question. I wish I had an answer for it. I think there's probably a lot of environmental cues that are going to trigger F plus cells to go out and find F minus cells. I'm going to guess maybe it's something like environmental pressure. So if there's some sort of, think about this from an evolutionary standpoint, you ask a really cool question is, what triggers this to occur? If you think about a population of cells living in an environment, and the environment's getting more challenging, more restricting, then they're going to, the whole idea of having new genetic information and recombination of genetic information is suddenly going to become very beneficial in the context of natural selection for those cells. The more genetic variation you have, the better. So I would think that would be the trigger. Yeah.
Would that have to do with like a quorum sensing maybe?
I don't know if quorum sensing, it's a really cool question. So, I don't know if quorum sensing itself is going to trigger conjugation. But that's something we could look up, and that's a cool question, because quorum sensing is definitely density dependent. It's a case where you have a higher density situation. They tend to quorum sense when they're in high density. They're much more likely to be somehow in a restricting environment when they're in high density. You know, resources are more likely to limit when they're in high density. But that specifically, if there's an actual physiological mechanism, a pathway for that, I'm not completely sure. That's a really cool question. Yeah, Yvonne?
Can you clarify the F plus and F minus group?
Yes, I can, and I haven't fully gotten to that. So she said, can you clarify the F plus and the F minus? And another really good question. An F plus cell has an F factor, okay? It's really this simple. And I'm gonna show you a pretty picture of this in just a second too. There is a chromosome. let's move on to the next slide, and that'll also help you understand that a little bit better too, and why it matters. So what happens is this F plus cell now has all the genes on that little plasmid for conjugation. So it can reach out and touch the F minus cell and give it its F factor. Alright. Okay, so let's step through the cartoon here. So here you have an F plus cell, it has a host chromosome, and it has an F factor, that autonomous little F plasmid that's in there. Alright, what it does is the F factor codes for things like the sex pills, and it codes for all the enzymes necessary to poke a hole in the recipient cell, etc. It codes for the genes necessary to replicate that plasmid. So the F plus cell reaches out and contacts the F minus cell via the sex pools. Okay? Then it wants to replicate that plasmid. It doesn't want to give it its plasmid, it wants to replicate its plasmid and give a copy of its plasmid. Okay. So the F factor basically replicates itself, and then it sends that replicating in a linear fashion, that replicating F factor down through the sex pellets into the recipient cell, such that now you end up with 2 F plus cells. So basically, this guy's got a copy of some music, he makes a copy of that music, he sends it down through the tube, and both guys have a copy of that music. Okay, so that's simple. In the old days, we used to burn CDs and stuff, but you guys don't do that. Alright, so point being, it creates a replicated copy so that they both end up being F plus. In the beginning, this was F plus and this was F minus. Alright, that is F plus, F minus mating. I use the word mating, but again, this isn't sexual reproduction. You guys are doing a great job not snickering and making funny jokes about all the innuendos. This isn't sexual reproduction, it's just horizontal gene transfer. They're moving their chromosomes horizontally, not vertically. Alright? It's about to get a little harder. Everybody comfortable with this? Got a lot of nods there, give me confidence. Okay, so let's make it harder. Oh shoot, super. Hooey. That's okay. Alright, so what can happen is you can have a cell that's actually an HFR cell. An HFR cell is an F plus cell. So, let's go back to our cartoon of this F plus cell. Here we have an F plus cell that has an F factor and a host chromosome. In some cases, that F factor, I'll do it dash, becomes linear and incorporates into the host chromosome. Same genes, same information, it is still an F plus cell, but now, because